Depending on which of my classes you're in, this slide might look familiar to you. I usually include it in the first couple of slides of the semester, right around the time we start talking about the scientific method, so real early stuff. And it includes Ian Wilmot, who is a scientist who was studying cloning in the 90s, and Dolly the sheep, who was a successfully cloned mammal. In fact, she was the first mammal of this size to be successfully cloned. So she represented a pretty big step forward in cloning things. We've been able to clone plants and other kinds of organisms, but Dolly the sheep was a big step forward in the field of cloning. The goal of science is to understand natural phenomena. So in these early few videos that we've been talking about in this playlist, we were talking about how cells differentiate, cell-to-cell communication, and how they turn on and off certain genes, all those different methods we talked about. And then the goal of technology is to take some of that information and apply scientific knowledge for some specific purpose. So now in the last video, we talked about nucleic acid hybridization, which is how we can see which genes are active or not active in a given cell type. We will talk about gene editing and gene therapy. And now we're going to talk a little bit about cloning, applications of our comprehension of how these things work. So, for example, biology is marked by discoveries like the discovery of DNA, the discovery of transcription, translation, how those mechanisms work, while technology is marked by inventions. However, these two concepts are interdependent. For example, the discovery of DNA technology, such as testing for hereditary diseases. For example, I've gotten some DNA checks myself, and I know that I am at an increased likelihood of developing what's called macular degeneration. It's pretty common for people who have blue eyes. My mom had it, my grandmom had it, so it wasn't that unexpected. It basically means that over time, my peripheral vision is going to narrow out a bit, but I seem to be clean for all other kinds of hereditary diseases that were being tested for, so 1 out of thousands is not so bad. And it's thanks to our understanding of how DNA works that I was able to get that kind of information. So this title says, Plant cloning shows that differentiated cells may retain all of their genetic potential. What does that mean? We've been talking about how every single cell in a body shares the same DNA, it has the same genome, right? But only certain genes are turned on and off. In order to make a clone, you need something called a totipotent cell. And that is a cell that can basically lose some of its differentiation. It's able to utilize any of the genes that are in the genome. So sometimes we can take a cell that is differentiated, that already is dedicated to a specific purpose, and we can induce it to return to the state of totipotency, where it can produce then every other kind of cell type as well. Let's get some definitions here. A clone is an individual organism created by asexual reproduction and thus is genetically identical to a single parent. Any cell capable of producing every kind of specialized cell in an organism is said to be totipotent. And the reason why we need totipotent cells is if I take like a skin cell and I try to clone it, it's going to do mitosis, it's just going to make more skin cells, right? But if we can find something that is able to then lose its identity as a skin cell and then become everything else, now we can build a full clone. Now, animals are able to actually do this. Some species are able to do this without the need for any gene technology through what's called regeneration. Regeneration is the regrowth of lost body parts, and it demonstrates that differentiation need not impair an animal's genetic potential. Basically, the cells can return to earlier states of development, and then because they're in that earlier phase, they can then be used to, say, regrow a limb. Not every organism can do this, and we are actively studying this process in other animals so that we can apply it to human medicine. How great would it be if we could regrow limbs from amputees or something like that? All right. I said cloning a plant is simple, and you would not believe how simple it is. So we go into a carrot, we get some of its roots here, and we shave off some of the root cells, and we put them into a test tube with a kind of growth culture medium. It's a liquid with a lot of nutrients and things like that inside of it. And this keeps the cells alive and allows them to continue to divide. And once they realize that they're no longer part of that whole carrot, they will have to return to a state of totipotency, and they will begin growing and forming a new carrot plant. So those root cells, some of them will differentiate into stem cells and leaf cells, and some of them will continue to be roots, and eventually you get a little plantlet. Isn't that a great name? A plantlet, which can then develop into an adult plant. And this carrot, since it's only been grown through mitosis from cells from another carrot, these two will be identical to each other. The new plant is a clone of the old plant. And you can do this with all kinds of plants. African violets, you can cut the stems off, put it in a pot of dirt, water it, and the bottom of the stems where you did the cutting will differentiate into roots, and eventually you'll get new violets blooming from that cloned plant. Animals are a little bit more complicated. The first thing we need is a cell that's going to be the cell that divides into our new clone. We can't have any of the original DNA inside of that cell. We got to get rid of it. So we're going to go in, we're going to remove the nucleus from that cell, and then we're going to implant the nucleus from a donor cell into this embryo that we're creating. So now we have an embryo with the nucleus from the donor cell added into it. That will go through the normal process of growth and development in vitro. So this is just like regular in vitro fertilization. We put it in a test tube, we give it nutrients, we put it under the right conditions, and it'll start dividing into a multicellular ball, a blastocyst, or a little bit later on, a morula. At that point, we can implant the blastocyst, the embryo, into a surrogate mother, in vitro fertilization, and then it will continue to develop to form a clone of the donor, the individual that donated this DNA right here being implanted in. Now, that's the idea in theory, but there was a few barriers before we were actually able to successfully do it. So in the process of animal cloning, called nuclear transplantation, sounds fancy, but nuclear as in nucleus, transplantation as in you're moving it, so it's literally called putting one nucleus into another cell, nuclear transplantation. DNA from a donor cell is inserted into a nucleus-free host egg, resulting in a clone of the donor. Now, what are the barriers I was talking about? Well, for one thing, we actually found out that we kept trying, and none of these embryos were coming to term. None of them were developing. And we realized that part of the problem was both the nucleus of the donor cell, the DNA in the nucleus of the donor cell, and the embryos being transplanted into had to be at the same phase of the cell cycle of mitosis in order for this to work. If there was a mismatch, then it threw everything off the rails and it just never divided and never continued. So in order to do that, they starved the cells. Sounds mean, but we're just talking about single cells here. They starve the cells of nutrients, which force them to go into a dormant phase, which we call G0. If you remember the steps of mitosis, you have interphase, which is G1, S, G2. Then you have mitosis, which is prophase, metaphase, anaphase, telophase. But when a cell is lacking nutrients, it stops the process of interphase and mitosis, and it goes into something like a resting phase, which we call G0. Basically, it's waiting to re-kickstart interphase again. So since we could induce both the embryo we were transplanting it into, the egg cell, and the nucleus into that same phase. Then when we put them in to each other, gave them some nutrients and gave them actually a little bit of an electric shock to get them going again. Now both were in the same phase, and that's turned out to be one of the keys to actually getting a cloned sheep like Dolly.